So, the latest Nobel Prize in Physiology or medicine went to solving one of the absolute biggest paradoxes in immunology. This is a story that kicks off with a massive question, follows two totally separate detective stories, and, well, it ends with a breakthrough that gave us the operating manual for our body's own internal peacekeepers.
So here's the big question, the one that bugged immunologists for more than a century. Think about it. Your immune system is an incredibly powerful killing machine. I mean, it has to be to protect us, right? But what's stopping it from turning on our own healthy tissues? What prevents what one scientist called horror autotoxicus, literally the horror of self-toxicity?
Okay, so for a long time, scientists thought they had this all figured out. They had a theory they called central tolerance. And for over 50 years, this was the prevailing dogma. The whole idea was that our bodies just deal with the problem right at the source, basically during boot camp for our immune cells.
The concept was pretty straightforward. You've got this organ called the thymus, and it acts like a training ground for your immune soldiers, the T cells. While they're developing, these T cells get tested. If a T cell shows it can recognize one of your body's own proteins, it gets flagged as a potential traitor and is immediately destroyed. Problem solved, right? For decades, everyone thought that was the whole story.
But here's the catch. It wasn't a perfect system, not even close. Researchers started to realize that no matter how tough the screening was, some of these self-reactive T cells always managed to slip through the cracks and get out into the body. So if you've got these rogue cells just patrolling your system, why are we all constantly battling autoimmune diseases? This little flaw created a huge mystery, and really, it set the stage for these Nobel-winning discoveries.
All right, this is where we get into the first part of our detective story. And it all centers on one researcher over in Japan who was willing to kind of swim against the scientific current, totally convinced there had to be another layer of control, an active police force for the immune system.
Enter Shimon Sakaguchi. He was really interested in this old and frankly discredited idea from the 1970s about something called suppressor T cells. That entire field of research had basically collapsed because the results were all over the place. But Sakaguchi, he thought the core idea was right. He was absolutely convinced that some cells weren't attackers, they were peacekeepers.
So what set Sakaguchi on this path? Well, it was this really bizarre experiment from the 80s. Scientists took newborn mice and removed their thymus. And what happened? The mice developed all these crazy autoimmune diseases. But here's the kicker. If they then took those same mice and injected them with a specific group of T cells from healthy adult mice, the diseases were prevented. This was the smoking gun. It proved that some specific cell population was actively putting the brakes on autoimmunity. They were acting like security guards.
Yeah, but finding these cells, that was the hard part. Sakaguchi spent more than 10 years trying to track down and identify this mysterious cell type. He needed to find a unique marker, a kind of uniform that would set the peacekeepers apart from the soldiers. And then in 1995, after all that work, he finally found it. So here's what he found. It was all about their uniform. We already knew about helper T cells, which have a protein marker called CD4, and killer T cells with their CD8 marker. Sakaguchi discovered his security guard cells were actually a type of helper cell. They had the CD4 marker, but they also had another protein called CD25. This unique combination, being positive for both CD4 and CD25, that was the key. He had just identified a whole new class of T cells, which he later named regulatory T cells.
Okay, so he's found his security guards, but the story is far from over. The rest of the scientific community, they remembered that whole failed suppressor cell idea, and they were really skeptical. He had found a link, sure, but he needed definitive, molecular proof of just how crucial these cells really were. And that proof, well, it was going to come from a totally different investigation happening an ocean away.
All right, put a pin in that for a second. We need to jump over to our second detective story. This one doesn't start in an immunology lab. It starts with a weird genetic fluke that was discovered way back during the Manhattan Project in the 1940s. Let's talk about the case of the scurfy mouse.
So picture this. It's the 1940s. In a lab that was studying radiation effects, some male mice were suddenly born with scaly skin, huge spleens, and a fatal system-wide immune rebellion. They were nicknamed scurfy mice. Researchers figured out pretty quickly that the mutation was on the X chromosome, but for decades, nobody could find the specific gene responsible. It was a total mystery.
Fast forward to the 1990s. Two researchers, Brunkow and Fred Ramsdell, decided they were going to be the ones to find that gene. Just to give you an idea of the challenge they were facing, they were looking at the mouse X chromosome.
That's about 170 million DNA base pairs. Finding one tiny mistake in all of that? I mean, talk about a needle in a gigantic haystack.
So how did they even do it? Well, they had a plan. Using the tools they had back then, they first narrowed down the search to a smaller region of about 500,000 base pairs. In that zone, they pinpointed 20 possible genes. Then came the really hard work of comparing every single one of those 20 genes between the scurfy mice and healthy mice. Gene after gene, nothing, until they hit the very last one on their list.
And there it was. On that 20th, that final gene, bingo, they found it. A tiny 2 base pair insertion that basically broke the gene. It was a gene no one had ever seen before, so they named it FOXP3. They had finally found the genetic cause of the scurfy mouse's immune mutiny.
Okay. And this is the moment. This is where our two completely separate detective stories just slam into each other and become one massive discovery. Brunkow and Ramsdell had the gene. Sakaguchi had the cell. Now all the pieces were about to fall right into place. So, the first thing they realized was a pretty big deal for us humans. The scurfy mouse condition looked a whole lot like a rare but really devastating human disease called IPEX, which causes fatal autoimmunity in young boys. So they tested patients with IPEX, and you guessed it, they found mutations in the human version of that exact same gene, which we spell FOXP3.
So let's just recap how this all came together, because it's pretty amazing. First, you have Sakaguchi, who identifies the security guard cells back in '95. Then, in 2001, Brunkow and Ramsdell identify FOX3 as this critical master switch gene. At that point, the entire global research community just went into overdrive. And by 2003, multiple teams, including Sakaguchi's, had proven the definitive link.
And suddenly, the final piece of the puzzle just clicked into place. FOXP3 is the master gene that controls the development and function of Sakaguchi's security guard cells. The scurfy mice and the kids with IPEX were sick because one faulty gene meant they couldn't produce these essential peacekeepers. This unified everything and gave us our modern definition of a regulatory T cell, or Treg for short.
So, you might be thinking, okay, cool science story, but this was so much more than just solving a biological puzzle. Discovering Tregs and their master switch, FOXP3, it opened up a revolutionary new chapter in medicine, the ability to actually, intentionally control our own immune systems.
See, what these Nobel laureates really gave us wasn't just an answer, it was a target. For the very first time, we had a molecular handle on the exact cells responsible for keeping the immune system in check. We basically had an operating manual for the body's own security guards.
And the way we can use this is really like a two-sided coin. It's fascinating. So in cancer, we now know that tumors actually recruit Tregs to form a protective shield around themselves. So the solution? Block those Tregs and you unleash the immune system to attack the cancer. On the flip side, with autoimmune diseases or transplant rejection, the immune system is way too aggressive. The solution there? Boost the Tregs to calm the whole system down and protect your tissues.
And hey, this isn't just some far-off theory. We're talking about over 200 clinical trials happening right now, exploring therapies that either block Tregs or boost them for everything from cancer and transplant rejection to autoimmune diseases like type 1 diabetes. This is the real-world impact of that fundamental discovery.
So think about where we started. A paradox that stumped scientists for 100 years, all the way to a molecular-level understanding that has totally changed immunology. We've gone from just watching the immune system do its thing to potentially being able to conduct it like an orchestra. And the question that leaves us with is, now that we have the manual, how are we going to rewrite the rules of disease?